Hello, thanks for joining me. I'm really excited to have our first live nerdy guest on Facebook's Dear Pandemic. I'm here today talking with James Conway, who's an infectious disease pediatrician at UW-Madison. He's a professor of pediatrics in the Division of Infectious Disease, University of Wisconsin-Madison School of Medicine and Public Health. And he's also the Associate Director of the Global Health Institute here at UW-Madison. And he's my collaborator and mentor. We work together on some vaccine stuff. So I'm really excited to hear Jim talk today about vaccine development and in a general way, and also the progress on the COVID-19 vaccine. So with that, I'm going to let Jim take it away. Oh, and I should say I'm also joined today by nerdy girl Lindsay Leininger, who's going to help me tackle some Q&A live after the video. So viewers, if you have questions, put them in the comment box, and we will try and get to as many of those questions as we can after Jim finishes his talk. So take it away.
Thanks for having me, Malia. Looking forward to talking about this. So I'm going to talk about vaccines in general and how they're developed, but specifically focus in on what's been going on and where we are with this whole need for a COVID-19 vaccine. Changing the slide. There we go. So just to give people a quick sense of the timeline that we're on, because one of the things I think people need to recognize is that generally vaccines are considered something that takes a decent amount of time, mostly about 8 to 10 years, to really go from conception to actually getting vaccine into people. And so I think people need to sort of take everything with a grain of salt, because we're really only about 6 months into this whole mess. So if you look at the timeline, patient 0 was in late December in Wuhan, China, when they noticed a atypical pneumonia case. And the very first confirmed case was early January of 2020. Within a few weeks, it was clear that there had been a cluster of cases at a Wuhan seafood market. And the estimates are that those first cases probably emerged sometime in October or November. And then that Wuhan seafood market then was closed in the beginning of January. First publications came out within a few weeks, and the New England Journal had the first one of a novel coronavirus patient in China in 2019, and actually had the virus isolated and sequenced through the program at Wuhan, where they have a really excellent virology program. First case outside of China was in mid-January. This was declared a global emergency, public health emergency of international concern by the WHO at the end of January. So for those people who think that the WHO was slow on the response, I think it's pretty clear that once they had information, they did take it seriously. They did wait to declare it a pandemic until mid-March, but that largely depends on the fact that you have to have multiple areas and multiple continents involved to really be truly considered a declared pandemic. But that's basically when I think people in the United States then became more aware of this, and that's when most of our states then started going into lockdown and shutdown. Interestingly, and I think one of the things that people need to be aware of is that we're in a very different era than we've been during past pandemics. The last one was 2009 with H1N1. And we're in an era where the internet makes things freely available, and science has actually become much more transparent. And so if you look at the cases of COVID-19 or SARS-CoV-2, and you look at the deaths, it almost directly tracks along with the number of journal articles that have been published, and more importantly, the number of preprint journal articles that have been put out. One of the things I just want people to be aware of is that the amount of science that's being generated, as this is sort of all hands on deck in dealing with this pandemic, has led to something that's, I think, unprecedented in the medical and scientific publication world. And that's this opportunity for science to be transparent and to not only publish rapidly in conjunction with what seems to be the rise of number of cases in the world, but also this ability to put things out even before they've been peer reviewed and decided that it was actually a bona fide, reasonable paper that's worth studying. And so about more than a third of papers that have been put out and picked up by the media and shared as sort of the latest, greatest thing are actually things that are in preprint stage that in normal circumstances, most of us would never even see. And I think this is one of the reasons that people need to be cautious about what they're seeing in the media, but also understand why it seems like there's this constant either pivoting or or changing of people's minds over various things because there is just so much stuff being generated. All right, so let's talk about this need for a vaccine. I think people recognize that we can't stay in isolation forever, nor can people maintain social distancing in a way that will completely control this. Testing availability, while it's getting better, still depends on contact tracing to try to get people to go into isolation. But to really be able to control a pandemic like this, and certainly like we did in 2009 with the H1N1 pandemic, will require a vaccine, and vaccines to be widely available to the point where we can actually achieve a herd immunity, a population of immunity, at least of 70 or 80% of the population. And so this is an all hands on deck activity, and I think people need to understand how this works. So first, let's just look at what it is to be immune to something. So we think about immunity in two different ways. There's innate immunity, things that you're naturally protected against, and then there's adaptive immunity, where you can either develop it naturally from having an active infection or receiving some of your mother's antibodies. If you're an infant, as an embryo, you get transplacental antibodies. So that's natural immunity. Artificial immunity is where you actually have the ability and exposure to something where you develop your own immune response to it, which is what we're trying to achieve with an immunization. Now, we can also give people passive antibodies, and so I think people are hearing about the use of COVID survivor plasma and the monoclonal antibodies that people are developing. That's another type of immunity that we call passive immunity. But the idea for a vaccine is essentially to try to mimic what an infection does in a way that actually stimulates the immune system. And the parts of the immune system we're primarily looking at stimulating are the B lymphocytes, which make antibodies. And so when people are hearing about antibody testing and serology testing, that's what's being looked for to see if people recovered from an infection. And T lymphocytes, the other group of lymphocytes within the immune system that are actually sort of the coordinators. These are the ones when you think about HIV and other things and you hear about CD4 cells and CD8 cells, that's what T lymphocytes are. And they're a really interesting group of lymphocytes that are actually capable, some of them, of killing things, including the cells that are infected or viruses, as well as the kind of the traffic directors within the immune system. And so what you want when you're developing active immunity is the ability to stimulate both parts of this so that you actually have the ability to remember and keep your immunity long term. Now, this is complicated, but this is just to give people a sense for all the different types of vaccines that we've worked over time in developing. Back in the old days, they used the entire pathogen, whether it was the smallpox virus or whether it was rabies or pertussis, the whole cell pertussis vaccine. This is the crudest version of a vaccine where you literally just take the organism, kill it, grind it up, and inject it into people. As we've moved forward with science, there's a lot more sophisticated ways that these are done. So polysaccharides, these are the sugar coating around a lot of organisms. These have been used to make some vaccines. Toxoids like tetanus and diphtheria that actually the organisms create a toxin. And so you can actually use that toxoid to create a vaccine that protects you against the toxins that those organisms make. And then we've gotten more and more sophisticated. We have split and subunit vaccines where we take pieces of viruses. We have reassortants where we actually create mutant viruses and put different things together. And then what was considered to be sort of futuristic type stuff has actually come to pass to be now some of the stuff we're looking at for COVID-19. And this is using vectors, in other words, certain viruses to carry other things, using mRNA or DNA plasmids. And all of these kind of structural vaccinology things are actually the things that many of these new vaccines are trying to leverage to create vaccines that can be used for COVID-19. But one of the reasons I think people need to be at least cautious about being too optimistic is that this is technology that's actually never successfully been used to make any vaccines. And most of the vaccines that you keep hearing about that people are so excited about are actually using this novel technology to develop a vaccine against an organism or a group of organisms that we've never actually successfully made vaccines against before. And so I think that's one of the reasons I want people to be cautious about what they hear in the media. All right, so then let's think about what is it that would be an ideal vaccine that you'd want to try to produce. So basically it comes down to two different things. Does it work and is it safe? So efficacy is what we talk about when we talk about does it work? Does it produce immune protection that actually can prevent infection? Ideally, that's going to be long lasting and easily boosted when you either get confronted by the organism again or when you get another dose of the vaccine. Ideally, not only does it protect you from getting infected and getting sick, but it also protects you from being able to be sick asymptomatically. In other words, being infected but not being actually having symptoms, and yet being able to shed the virus. And so we're hearing a lot about this lately, that people are trying to figure out how common is it that people can actually not have symptoms and yet be able to infect other people. So an ideal vaccine will not only protect you from getting infected and being sick, but protect you from being infected at all and being able to shed and transmit to others. And then, as I mentioned before, we want it to be able to activate both the T cell and the B cell side of the immune system. From a safety standpoint, we want to know that if you then later get confronted by the organism and get infected, that having been vaccinated doesn't lead to you getting worse disease than it would have if you had not been immunized. And while people say, well, how could that happen? This has actually happened multiple times with vaccine trials in the past, most recently with a dengue vaccine that was introduced into Southeast Asia, where it unfortunately turned out that when people then got certain types of dengue after getting the vaccine, they got much worse disease than if they hadn't been vaccinated at all. And there's been certain vaccines through history that were developed and then never introduced into the public because that was in fact one of the bad side effects that happened. You also obviously want to minimize what side effects happen to people after they get the vaccine. Certainly it can hurt and give people swelling and pain and maybe fever, but you don't want anything worse than that. And then obviously you want to know that there's no long-term side effects from getting that vaccine. Here's what these coronaviruses look like under an electron microscope. These are obviously cartoon versions of these things. And so when you're thinking about making a vaccine, what you want to do is try to find things on these organisms that are conserved. So even though these viruses constantly mutate as they replicate and get transmitted from person to person, you want to find parts of the virus that are pretty stable and that are readily presented to the immune system, so they're pretty visible. So most of the vaccines that you're hearing about are directed towards this spike protein. The reason coronavirus is called coronavirus is it looks like a crown, and the crown is basically made up of these spike proteins that are involved in the attachment of the virus to cells that then allow infection. So most of the vaccines you're hearing about are related to using spike proteins, but there are a whole lot of other pieces of these things that could be potential candidate sites for a vaccine in the future. All right, so then let's think about what it's going to take to actually develop a coronavirus vaccine and do it in a short period of time. So first off, we have to recognize that coronaviruses are very, very common. In some studies that we've done here in Wisconsin, about 10 to 15% of colds that people get in the winter are caused by coronaviruses. But we know that similar to other cold viruses and even flu viruses, that natural immunity to coronaviruses doesn't last very long. And so you can get colds over and over every season. Just because you got a cold one year doesn't mean you're immune to colds in the coming winters. And unfortunately, that seems to be pretty consistent for most coronaviruses. So we already know that even if you get infected by COVID-19, we have concerns that that immunity may not actually be long lasting. So that's concern one, is can you actually make a vaccine that's better than the natural immunity you get after the infection? Secondly, we're not even sure what the markers of immunity are. I mean, people are measuring these antibodies against the spike proteins, but we don't even really know how well those actually act over time in protecting you from future infections. And so one of the questions you have to answer is that we don't even know how to measure immunity using a blood test. Maybe the only way we can actually decide whether these things work is by having people exposed to the actual virus and see if they get sick. Now, there's two ways that can happen. One is it can happen just naturally if you're in an outbreak area where the virus is circulating, people are naturally getting exposed. That's a great way to figure it out. Unfortunately, most of the places that are actually working on developing vaccines are the areas where the curve has been flattened and cases are actually decreasing. And so scientists are concerned that a lot of the people that are starting to be tested for the vaccines are in areas where there actually isn't a lot of virus circulating. So the other question is then, would you actually ethically be allowed to and consider it reasonable to intentionally squirt COVID-19 virus into people's faces who have been vaccinated? Because that's being proposed and discussed as a way of trying to fast track these vaccines. As I mentioned, figuring out what are the best antigens, what are the best pieces of the virus, what do we do if these things start mutating? What's the best route of delivery? You know, we have lots of different ways to give vaccines now. We have intranasal vaccines and oral vaccines and injectable vaccines that go into muscle or go into the skin. So what's the best way to get the immune system to see these? I mentioned the enhanced disease. That's going to be something that needs to be understood. How long do these last? Is it ethical to challenge people with these viruses? Who do we test these in? You know, most of these vaccines primarily get tested in healthy males 18 to 45, because people don't want to experiment on women of childbearing age who could get infected. People don't want to experiment on old people or young kids. And most places and scientists don't want too many variables. And so you don't want people with a whole lot of other comorbidities, a lot of other medical conditions that might skew your results or make things confusing. So these things are mostly tested in healthy young males, often in many populations, white males, in a situation where our target population, if you look at the highest risk people for having bad outcomes, it's people over 60, people with comorbidities, and in many areas of the world, we're seeing it really most devastating in communities of color. And so the idea of testing it in one population to then use it in a completely different population does make people a little squeamish. And then I'm going to talk a little bit about the progressive progress and the process for making a product like this. And then obviously the question is, can we mass produce these and get to the point where we've got 8 billion doses if we want one for every person on Earth? But most vaccines actually require multiple doses, so maybe 16 billion doses or 24 billion doses, which we've never ever done with any vaccine ever in the history of the world. That's the depressing part of it. Okay, let's get to a little bit more optimism. So first of all, this is an all hands on deck. This stuff is happening all over the world, and you can follow this on an open crowdsource site, vaxmap.org, where literally every place on the planet that is involved in either research or developing manufacturing facilities is placed on this map. And you can see that this really has been a worldwide effort. There's also a lot of candidates in the pipeline that people have been looking at. You know, in the vaccine world, we usually talk about the fact that about 1 in 100 vaccines that people try to make happen actually make it to the point where they're used in humans. Well, looking at the databases that I follow by the WHO and Milken Institute, there's already 126 preclinical vaccine candidates that are registered at WHO. And in fact, the Milken Institute has an additional 7 that are also in preclinical development. And these are all tackling options for vaccines from totally different angles. So you've got DNA-based vaccines, you've got inactivated virus vaccines, which is the model we use for a lot of the current vaccines that we have for kids and adults. There's non-replicating viral vectors, where they actually take other viruses, put pieces of COVID-19 into it, and then use that virus to deliver the pieces to stimulate the immune system. There's protein subunits, there's RNA vaccines that you've heard about, where these mRNA molecules get transcribed and translated into proteins for the immune system to respond to. There's virus-like particles, which are like the HPV vaccine. And then there's actually live vaccines, replicating virus vaccines, like the nasal flu vaccine model. There's live attenuated viruses, like we do for measles and mumps. And then replicating bacteria that actually contain pieces of the virus as a way of delivering viral material. So there is a lot of stuff in the pipeline. I won't go through all of these, but these are the 10 current, as of yesterday, or as of 2 days ago, I guess, virus vaccines that are currently listed at the WHO site. These are the 10 candidate vaccines. And you can see there's 3 different inactivated vaccines. Those are pretty classic versions of vaccines that would be likely to be able to get to market soon because we've done those before. But you can see that there's a lot of these non-replicating viral vectors or RNA vaccines, or even a DNA vaccine. There's technology that's never been used to create a vaccine that we currently use, that are all in various stages and trials. The other thing I think that's very interesting and encouraging is that you're also seeing some really novel collaborations between all sorts of different companies. Some companies that are good at growing viruses, some companies that are good at making adjuvants, which are chemicals you can put into vaccines and stimulate the immune system a little bit better. Some companies that are just really good at making stuff in large quantities. And so you're seeing these collaborations come together that at least I think are giving people a little more optimism about how this might work out. Now let's talk about the phases of how you actually develop a vaccine. So the traditional way of developing a vaccine is a bunch of scientific nerds hang out in a lab for a long time trying to figure out how to make something, test them in animals to see if it actually even proves the concept. Then you start out with healthy individuals, just a few, to figure out what the right dose is. You go then into hundreds of people to look much more for these safety signals and make sure things actually look like they're safe, as well as starting to think about how do we actually look at whether these things really work, which is when you get into phase 3 trials. So all of these vaccines that I just mentioned, when you look at where they are in their trials, they're all basically in phase 1 or phase 2. in the traditional development program. So they have yet to be introduced into large populations. Once it gets to this stage where you have enough people to know, are there going to be any rare side effects and does it really work? That's when it then enters into the general population for broad use. And I think one of the things people should be reassured by is it's not like suddenly the watchdogs are called off when it's introduced. And in fact, this is actually where more vigilance goes in once these become broadly used to make sure that there aren't any rare side effects or unusual things that happen. So there's a constant surveillance process that happens in every country where vaccines are used. Obviously, ours are through the FDA, but every country in every area of the world has something similar. So when you look at that traditional means of going through a vaccine, whoops, sorry about that. What you see is this sort of very stepwise staged process before you get to the point where it's large scale licensure and then large scale manufacturing. And really the manufacturing idea doesn't start until you've got pretty confident results from phase 2 into phase 3. Okay, this looks like it might be a go. Let's get this ramped up. What you're seeing with Operation Warpspeed and other programs around the world is this idea of developing COVID vaccines at pandemic speed. This is an article that came out in the New England Journal a couple of weeks ago that basically summarizes this process. And what the new approach is basically what we're calling stacking, where you're essentially getting all of these things happening all at once. You're getting scientists coming up with ideas. You're getting almost immediately those being put into some kind of clinical development in these phase 1 and phase 2 testing at the very same time that they're actually already ramping up and building manufacturing capacity, getting vials for transportation and all the cold chain things worked out so that as soon as there's something that's a decent candidate, those can go into large scale manufacturing. And so this is why people keep talking about maybe we can pull this off in 12 to 18 months as opposed to the usual 8 to 10 years because we're doing it in this stacking mechanism. I'll finish up with just giving people a sense for what the US program is. I know we've got people probably from all over the world, but the way Operation Warpspeed has been crafted by the US government is basically having looked at the most promising candidates from those 100+ candidates that I mentioned to you that were currently in development, kind of whittling it down to 8 that seem to be the furthest along, and then taking it down to about 3 to 5 of these. Looks like there's going to be about 5 that the administration has chosen that are going to go into ramped up production as well as randomized trials and trying to figure out the safety and efficacy stuff as quickly as possible. And so this is basically the process we're in. As far as manufacturing, the government's investing an enormous amount into manufacturing and distribution, with the government taking the risk to do that. You know, if you're a private company, you're not going to go build a whole new plant and risk it unless you know that you've got a candidate that's looking really good in phase 3. Government is taking an enormous gamble by actually building production facilities to make some of these vaccines, even before we know whether these vaccines are safe and whether they work. And then the idea is then this is going to be advanced into rapid development and scaled up immediately after the authorization or approval process happens, so that then these can be used as quickly as possible and distributed as widely as possible. And there are going to be some agreements as far as where these are going to be distributed to make sure that there's hopefully some equity into who gets access. Because this distribution process, I think, is the part that makes some people nervous. You know, this is an unprecedented effort to try to get hundreds of millions of Americans immunized in a timely manner. You know, currently the industry makes about 170 million doses of flu vaccine every year, and about, you know, that covers about 40 to 50% of the population of 330 million. You know, the task is basically developing enough for not only all of America, but all of the world, and likely needing multiple doses. So Operation Warpspeed is basically focusing on all aspects of this, not only just the production, but also how do you store these things, how do you transport them. And the Department of Defense is actually going to be part of this process then, because they hope at least that their distribution possibilities are going to be better than what the current private medical infrastructure would allow. They're committed to affordability, and there's this whole agreement that's supposedly in action that there's going to be a donated allocation so that these are hopefully going to be equitably available. So financially, we're looking at about $10 billion through the CARES Act that has been dedicated to this. There's another $6.5 billion that's gone to the Biomedical Advanced Research and Development Authority, and then about $3 billion that's gone to NIH to support these research programs. This is, again, is an unprecedented investment in something like this, and the reason that I think people have at least some optimism. Last thing, just recognizing, obviously, there's a lot of opinions out there, and there's a lot of distrust of science and of the pharmaceutical companies as well as of the government. And I think that that's real, and I think we need to be aware of it and address it up front. This is an interesting study that came out of the Associated Press Study Center at the University of Chicago. And just a couple of quick looks at how people responded when they were asked about these vaccines. So the first question was, do you plan on getting vaccinated against coronavirus? And overall, only about half of people said that they were definitely going to get vaccinated. If you look at the distribution, it's pretty evenly distributed over where people's thoughts are. We do see that in populations of color, there does seem to be some distrust and lack of eagerness to embrace these kinds of vaccines. These seem to be skewed pretty widely across political bases, but you can see that there's certainly hesitancy in the general population about accepting a vaccine, especially one that's being brought to market in a much more speedy fashion than usual. When they asked the subgroup who said they would want to get vaccinated, why? Obviously, the vast majority want to protect themselves and their families and avoid getting seriously ill. People also talk about the fact that it would make them feel safe around other people, that they would contribute at least to the protection of their community, and they don't think life will actually go back to normal until we get broad-scale immunization. On the flip side, when you ask people why they wouldn't get vaccinated, the biggest thing is obviously concern about side effects. People have concerns about being able to get infected by the vaccine and actually causing COVID-19, which is some of the same concerns we see with people that refuse flu vaccines. Good portion of the population isn't concerned about even getting seriously ill, or may not think that the vaccines work well, or just don't think that the outbreak is as serious as it seems to be. And so we're going to make sure that we address those. Those are going to be things that are going to have to be important in the messaging and the discussions about this. So I'll finish up just reminding people that in the meantime, until we do get a vaccine, remember this is a person-to-person transmitted virus. Please stay home if you're ill with any symptoms that could resemble any kind of respiratory illness. Hand hygiene and keeping your hands away from your own face, maintaining physical distance from others of at least 6 feet, wearing a mask at least when you're in confined public places, both for your own protection and protecting others in case you're one of those people that has an asymptomatic infection. And then finally, if you do nothing else, get immunized against flu this year. COVID-19 and flu appear and act very similarly. And so influenza-like illnesses are how these things show up. Our emergency departments are already overwhelmed in many places. And when flu and cold season shows up, it's going to be that plus COVID-19 on top of it probably. And so anything we can do to get flu to circulate less will make the ability to deal with COVID-19 that much easier for everyone. So in the meantime, the whole world's wearing masks. And I think it makes sense in this current climate to do so. And with that, I'll close down and see what people have questions about.
Yeah, thanks so much, Jim. That was terrific. We got a couple great questions in the comments already. And for those of you who are watching, feel free to put your questions in the comment box and I will, we'll get to as many of those as we can for Jim here, but I'll just start off with one. Can you tell us, so we get a lot of questions about this idea that the vaccine development process is proceeding too rapidly, that we've skipped animal trials, and that's a concern. What are your thoughts about how fast this development is proceeding in terms of its safety?
Yeah, I mean, that's the million-dollar question. I mean, I think in most circumstances, the feeling I have is that we're not cutting corners so much as just going through things in a much more rapid fashion and doing a lot of things simultaneously. I do feel I'm appreciative of anybody that's willing to be in these trials, because without people volunteering to be candidates for these trials, we wouldn't be able to do the things we do. I think that the one part that is going to be challenging is the efficacy and understanding what happens when people are challenged by virus directly being thrown in their face. And that part of the immune response, I think, is going to be really important to sort out. But I understand, I share people's concerns. I mean, that's one of the reasons I'm willing to talk about these things and be transparent about it, is that this is unprecedented. And I do hope and expect that the regulatory authorities, even with the political pressure that they're under to get these things to come out quickly, are going to stick by their guns and require each of these groups developing these things to still present the usual data that is needed. The good thing is that I think that the transparency of all this science has not only allowed the public to be watchful of these things, but allows all of us that are scientists and are involved in vaccine work also to be scrutinizing all this stuff. And I think you're going to see us be the ones, as much as anybody, to call out the government organizations that are doing these things and pushing back on them if we do think that they are pushing things too quickly and not really measuring safety in the way we think it's appropriate. Because, not only is there danger to doing this in a way that doesn't protect the public, but it certainly endangers confidence in vaccines as a whole. So, you do have to maintain standards no matter how much we need these vaccines.
Yeah, absolutely.
I have a follow-up, if you don't mind, Malia.
Yeah, go for it.
So it's something that you had said, Jim, that has pinged a question that we've seen from our followers, and I've had myself. So there was a really interesting long-form piece by Helen Branswell in Stat News where she interviewed Dr. Fauci about his thoughts on vaccines, and perhaps you've seen it. But one piece that I think has been quite salient has been his communication that his big concern regards efficacy, that he's very worried that this vaccine, he was not so much highlighting the safety concerns, but rather the efficacy concerns. So I was hoping maybe you could explain for our followers, for lay people, what those efficacy concerns are.
Yeah, so I think Tony's comments, he's done a phenomenal job of trying to message this in a way that's understandable for people. I think that when you talk about efficacy, you're essentially saying, how well does it work? And I think the closest model that we have that we're seeing for a vaccine are going to be related to the flu vaccines. And so when we see flu vaccines, you see every year the media jumps on certain efficacy numbers that say, oh, it's only working for 9% of people and things like that. And one of the things you have to remember about efficacy is that the world is not black and white when it comes to vaccines, but yet that's how a lot of the data is generated. So when you say that the flu vaccine on the worst year I remember was only 9% effective, that means it was 9% effective at completely preventing any disease. What we often talk about for a lot of efficacy is, does it at least prevent hospitalization and death? And so even if you look at the year that the flu vaccine was only 9% effective, in fact, that year it was about 65% effective at preventing severe disease and hospitalization. So when you talk about questioning efficacy, you first need to decide, what are the benchmarks we're even trying to achieve? And I think the benchmarks we're trying to achieve is preventing severe disease and hopefully preventing transmission to other people. The other piece of efficacy is then, how long does it last? And so, while he out of one side of his mouth says, he's optimistic that we'll have millions of doses available by early 2021, he also says, but we're going to need to study the efficacy and understand how well does it work and how long does it work. And I think one of the things we're going to probably see is that there's going to be an initial wave of vaccines that come out that are probably okay. They're built on the inactivated model of how flu vaccines currently are developed, and they'll work okay. They probably will blunt the curve a little bit and maybe help protect people from getting severely ill. But the rest of the science is still going to keep marching on to try to develop something that really, really works well. The other part of the efficacy, though, is efficacy also means that you're not potentiating, that you're not actually causing worsening of anything. And I think that's the part that he's most concerned about. I think many of us are concerned about. If you think about, people have probably heard about these COVID toes or this multi-system inflammatory disease that children get. This is really interesting because these are kids that get COVID-19, often get such a mild version, they don't even have classic symptoms. And then 4 or 6 weeks later, they have this inflammatory response that essentially their immune system goes overboard and attacks itself. And so, we know that that's a thing in kids. And so people become nervous that, is there anything with these vaccines that could stimulate that same kind of response and actually do something like that? So that's a long way around saying that efficacy is complicated, but you have to set, currently I think we have a pretty low bar that we're going to get over. We just want to know, does it protect people from getting really sick, and does it at least limit the amount of transmission they can make? but I don't think it's going to be the perfect vaccine for a while.
So will
So
there be multiple vaccines? That's
I
a
was just
question.
gonna ask that same question.
On the Facebook chat.
I fully expect it's going to be like flu. You know, currently we've got, I can't even keep track of all the flu vaccine manufacturers and all the different versions of flu vaccines that are out there. You know, there's at least 10 or 12 different ones. There's live ones, there's adjuvanted ones, there's high potency ones for the elderly. There's, you know, there's all these different versions of flu vaccine. I would expect that, you know, we're going to see of these 10 candidates and others that are going to enter trials, that many of them are actually going to eventually reach the point where they're mass produced and delivered. The other thing you have to recognize is that, you know, the whole world has gotten together at the World Health Assembly recently and agreed that whoever comes up with a vaccine is going to share the technology with other countries. The US actually chose not to participate in that. And so we may end up with whatever we've invested in for our population, and the rest of the world may end up with very different vaccines based on what is developed in Europe and China. But I would absolutely expect that there's not going to be a single vaccine, that there's going to be a whole bunch of different ones that are going to all have different safety profiles and probably different efficacy.
So we got a question related to what you just said. Somebody asked, do other countries have similar programs to Operation Warp Speed, and how might that speed up the vaccine availability to all corners of the world?
Yeah,
you kind of addressed that, but
yeah, the Chinese clearly have, you know, put a lot of money and a lot of investment into their particular vaccines, which is one of the reasons they've got a whole bunch of candidates already entering into phase 2. And then the European Union has collaborations. You know, there's also other organizations that have gotten involved, including PATH and Gavi and a bunch in the UN. So there's a lot of different collaborations happening all over the world. And it'll, you know, I sort of think about it as sort of like a cross-country race. You know, you start out with like 100 people in a pack running, and then eventually some of them are going faster than others. And so you're starting to see separation currently, but it doesn't necessarily mean that those ones that are out in front are going to be the winners. You know, they may get tired or may fall on their faces, and something that's still back in the pack may actually be the one that ends up being the home run that we're looking for. But I think initially you're probably going to see a whole bunch of different products, some being used in various areas of the world, others that become more universal.
So one of our followers asked, what's the rationale for the US opting out of that international cooperation agreement?
That's a great question. It largely falls
probably a question for a different kind of nerd.
Yeah,
right.
it largely falls in, I mean, no, it does largely fall into the America First mentality of the federal government currently. And so, you know, they've decided it similarly to, you know, threatening to completely remove themselves from the World Health Organization, that this is going to be an America First moonshot type. And there's been actually, I had a couple slides about that in here that I removed, but you know, there's been a couple of great articles written about the diplomatic side of this and how this is becoming a nationalistic thing. The Wall Street Journal had a great article a couple of weeks ago about the nationalism of vaccines that we're starting to see. So you're putting all your eggs into the baskets that you've got, and it'll be really interesting to see what happens if something else comes up that actually looks better, and what negotiations happen to make sharing happen with that technology.
Yeah. So a couple of related questions. I was wondering, how did we narrow down those most promising candidates? What makes them the most promising? And then there's a related question from a viewer. Do you have a particular vaccine candidate that you are most optimistic about?
So I'll answer the first one, and I'll contemplate whether I'm going to answer the second one. The ones that are actually, and it's a great question. The ones that are currently farthest along are largely based on vaccines that people had already been working on. Because if you remember, we've had coronavirus issues before. I mean, the original SARS, obviously, but even this MERS disease that went around, those were all coronaviruses. You know, coronaviruses have been considered a threat for decades. You know, we know there was even likely a very large coronavirus outbreak way back in the 1890s that was originally called the Russian flu. But in looking back at it historically, it meets many of the same criteria and probably was a coronavirus. And so coronaviruses have been a group of threats to humanity that groups have been working on vaccines for 20 years and haven't really had great success. But a lot of the candidates that you're seeing, the Oxford vaccine and some of the Chinese vaccines, already were kind of in development. Many of them had just been shelved when SARS kind of went away on its own, and then have been woken up again as money's been thrown around at these things. So a lot of the ones you're seeing were basically things that already had been partially in development for other coronaviruses and have just been retooled. Basically what they've done is taken the genetic material from this virus and cloned it into the existing vaccine that they had. And so lo and behold, they were already ready to go into clinical trials. So by no means should anybody imagine that just because those are the 10 that are out there and the US government has selected 5 of them, that those are likely to be the best ones. I think those are likely to be the first ones. And so that's why myself and Tony Fauci and others often talk about, the first stage or the first phase of vaccines are going to be okay, are going to probably give partial immunity and maybe short-lived immunity, but it's going to take time to get to something that really gives robust long-term immunity. What do I think the likely candidate is? I'm honestly not sure. We've been looking for I think everybody recognizes it's a pain in the neck or a pain in the arm to get a flu vaccine every year. And so, one of the holy grails of vaccine science has been developing a universal flu vaccine that you only need to get like every 10 years, like you do for a tetanus shot. And so, I think that there's so much attention and so much money being thrown at vaccines. I do have some optimism that maybe there actually could be a combined universal flu and coronavirus vaccine that you would at least only need to get periodically. There is one version that I'm relatively optimistic that's actually a collaboration between some researchers here at the University of Wisconsin, working with a biotech group at Bharat Biopharmaceuticals in India, which actually is a combined live flu and live COVID-19 combination vaccine that would be probably delivered intranasally like the current flu mist vaccine. And that actually, from a physiologic level, kind of mimics what the natural infection actually does, which is it enters into your nasal cavity, invades through there, and then that's where your protection needs to be. And so that's one of the reasons that may be a really interesting model that I would think actually has some high potential for being useful. But in this 140 odd candidates that are out there, there's some really novel technology that's never been used before. And so I'm holding out hope that there's something really game changing in there.
Great. Lindsay, you have any final questions? I think we're going to wrap up.
I do actually, if it's okay to pivot, there's some language that you used, Jem, that I really liked that I just wanted to be sure to amplify. And that's this transparency of science, that we are seeing the floodgates open of science that often has been behind the ivory tower before it makes it out into the public. And I feel like our followers and we all struggle with this whiplash effect that you also mentioned. So it'd be great if just to hear your take on how you handle the whiplash, right? Like when you get excited, when you caution people to slow down.
That's a great question, Lindsay. So I think there's a couple of things that I teach my students and learners and always keep in mind. One of the things I joke about in the medical and scientific literature, for every paper, there's an equal and opposite paper.
Yeah.
So you can almost always find something that supports almost anything that somebody's published along the way. Because I think one of the things we've had to recognize is that the scientific literature, just the volume of journals that are out there, just like every other field, has just exploded. I mean, there's all these open source, open access journals now. And so it's very rare that you ever run into a scientist or a medical person who can't find somewhere to publish something. Gone are the days where you'd submit something and get rejected, submit it a couple other places, get rejected, and then just give up. Eventually, somebody will publish anything at this rate. So I think we do need to be very cautious about what's out there. What I share with everybody is that until it's been reproduced over and over and over again, it doesn't become a truth. It just becomes a hypothesis and a theory that somebody's at least designed an experiment or has found a set of data that supports their hypothesis or their theory. But until there's reproducibility, where it can be done in different ways and get the same result, and where the method that they used can be replicated by another group and get the exact same thing to work the same way, I don't think that it's valid. And so there's a lot of things that kind of come floating across my listservs and everything that I just take note of as, that's interesting. And until I see it in 10 or 15 reproduced articles and from a number of different scientific sites, in my mind, it's almost no better than just somebody's opinion. And so one of the things that even the really high level journals have had to deal with is this rush to try to be the first ones to get things out there, and yet the trust that goes into the review and refereeing process. And so I think people, saw both the Lancet and the New England Journal of Medicine, both within a week in unprecedented nature, having to retract articles that people had been making a lot of policy decisions. And I had, with both of those articles, basically said, yeah, it's just one group of people with using the very same set of data, and they're publishing almost the same thing in two different journals. That's not reproducibility. That's actually just duplication. So until other groups could actually get that same finding, it didn't really mean anything to me. And to be honest, I had said it to a couple of my colleagues, I'm really surprised this got published. There must be more of these examples already out there that I just wasn't aware of. And then a few weeks later, the articles get retracted because in fact, they couldn't be replicated. And there was in fact some concerns about the source of the data. So I think reproducibility is key. And then I think obviously the impact factor of what the journal is that things get published in matters a lot. So when we look at scientific literature, how many times that article or that journal gets cited by other articles adds what we call an impact factor. It becomes a much more credible source of information. And so the first thing I do whenever I see something that's just different from what we think we know is, what journal is it? And then can I find anything else that substantiates and reproduces that finding? If those two criteria are met, then I'll give it the time to actually believe it. Otherwise, it just goes into a file of interesting things. And until I see more of them, it doesn't really become something I'll act on or take into consideration.
Terrific guidance. Thank you.
Yeah, I love that point. I think it's, you led with some of these questions about just the avalanche of information that we're trying to sift through, and how it seems like one day it's, one day we're headed east and the next day we're headed west on lots of different issues. And so that is really important to keep in mind, that it's an evolving science.
Yeah, I think the media is, obviously this is all that's on anybody's minds, until recently, at least with a lot of the recent tragic events. And so I think it's, even listening to like news radio or picking up a newspaper, like every page is something about COVID. And so you also get a little bit tired of it as well. And so I think the media is constantly looking for a new sparkly object to wave in front of us. And so they do all en masse jump on things. And, when those things get walked back, that's not such big news. And we've seen that over and over again, whether it's quotes from our president or people at WHO or articles that get published and retracted. Once it gets out there, that was the interesting part about it. When it gets walked back and discredited, that's not as big a news item. And so it's been unfortunate that some things that really were clearly not true continue to exist in the ether and on the internet and continue to propagate themselves. And so it has made things a lot harder. But I would encourage people, be well informed, but take everything with a grain of salt until you've seen, and it's not just 17 news articles have all said the same thing, but seeing that there's been multiple reproducibility from the sources of where they got their information, that's the only stuff I think that justifies attention and change in policies.
Yeah, absolutely. I think that's your mic drop, Jim.
Absolutely.
My earphones drop.
Yeah.
Okay, well, we will wrap there. Thank you so much, Jim, for joining us. This has been really informative. And to our viewers, we will post this to watch later on the page. I will try and stitch together the multiple false starts that we had getting the Facebook live stream going so that you can watch it all in one go if you want to. And yeah, thanks to Lindsay for helping me out with the Q&A. And thanks so much for watching Dear Pandemic.